Hello, everybody. This is Chunan Chen. As Allison already introduced, I am the co-founder and CEO of Single Cell Technology. And I want to start with a story. Several years ago, I was talking to someone at a major pharmaceutical company, and this is the time that we obviously try to offer our service technology for them to screen antibodies. So I asked the question, so what do you want? And the question is really, how do you want to screen for the antibodies? And the person said, I just want one antibody, just that one, that elusive one antibodies. So that question sort of stuck with me for a while. And because as a sort of the technology provider, we kind of wrapped up in screening for antibodies. And I guess it's maybe a subtle point. I forgot the point that everybody is really looking for that one antibody. So imagine in your hand, you have the next generation of Humira. What would you do? I think you would be probably happy, but you may not know until later on. So I think what I want to do today is to see that if our technology can help everybody get to that one antibodies, and we'll see how we do here. Here's the outline. We're going to talk about our technology in the sort of context of a demonstration project. And the reason of doing a demonstration project is that way we actually have more sort of freedom to talk about the data and actually to do the things, some of the projects that we wanted to do instead of a customer's projects, which is something that we also do too. And then for that, we actually selected the BCMA as a target, and then we talk about the experience of doing that and the lesson we learned during that campaign. And then after that, I will use the opportunity to introduce Abtheneum, which is our antibody discovery platform, and some of these salient features. And as I talk about the features, I want to talk about the implication of some of these features as it pertains to screening for antibodies. And later we'll talk about what's in development, and I'm going to close. And then let's go on. So as I talk about this demonstration project using BCMA, what we wanted to do is to really look at some other aspect of the antibody screening. Here we actually wanted to look at the challenges immediately upstream of our technology, and that includes protein handling. And the reason is when we do customers' project, the protein antigens are just handed to us, and everything is done, or sometimes it's handed to the animal service provider, and we never see the antigen. So we want to do some part of that, and obviously we receive the protein for labeling purposes, so we do handle proteins. But we never really got involved in the immunization aspect of it when it pertains to screening. So we want to do something like that. And so we want to look at how the immunization and how it impacts our screening. So this is one of the reasons of doing this demonstration project. And this is also the time that we can pick some of the antigen that we are interested in doing. And this is also a time that we can reconstruct some of the antibodies we find and then see if we have any sort of predictions where we have some thoughts about these antibodies based on the sequences of these, and to see if there's any pattern we can find. And obviously this is going to be an ongoing studies. And at the end of this project, we'll end up with some antibody that may have interesting properties, so that will serve as a asset for us, and that will create values for single cell technology. And then along the line, we'll be able to showcase the features, some of the strength of Abtheneum. And by the way, we're using the high quality proteins from AquaBiosystems. And this is the case that we can also showcase their protein portfolio. And so that's the overall project objectives. And let's go into BCMA a little bit. I'm not going to talk about a lot because I'm pretty sure everybody's familiar with it. It's a member of the TNF receptor superfamilies and expressed in the mature B lymphocytes. And actually, I think in the case of the multiple myeloma, a lot of them are expressed almost exclusively on the malignant plasma cells. So that makes it an ideal target for